So it's my pleasure to introduce our first speaker, Andrea Gore. Dr. Gore is a professor at the University of Texas, where she's the chair of pharmacology. She'll be speaking to us today about her very interesting work on the inter and transgenerational effects of endocrine disrupting compounds on the developing brain.
I'd like to thank the organizers for inviting me to speak on my research. I'm very excited to be able to start the session on transgenerational effects of endocrine disrupting chemicals, or EDCs, on the developing brain. And I'd like to begin by acknowledging all of the people who have done an incredible amount of work on intergenerational studies that have gone all the way through the 6th generation. It's a real labor of love, and so I wanted to have everybody's picture up. I'd also like to thank the National Institute of Environmental Health Sciences that has had the wisdom to fund intergenerational research on EDCs and has been the sponsor of the work that I'm about to speak about today. Okay, so my lab is interested in effects of EDCs on the developing brain. And the brain is not the typical target that we think of when we talk about endocrine disrupting chemicals. But in fact, the brain is one of the most hormone sensitive regions in the body. We know that exposures to hormones early in life organize brain development, and this leads to both structural and functional changes that are sexually dimorphic, in other words, are different between the sexes. And the reason that this happens is because in the fetus, the male testis produces quite high levels of testosterone, whereas the fetal female ovary is basically quiescent and does not produce much in the way of hormones, either testosterone or, for that matter, estradiol. And in addition, in the brain of males, because testosterone from the testis can get into the brain, much of that testosterone is converted into another hormone, estradiol, through actions of the enzyme aromatase. When we think about the brain of a fetus, then the male brain is exposed to higher concentrations of both testosterone and estradiol. And those sex differences are critically important in shaping the development of the nervous system. Disruption of these natural hormone actions by EDCs can perturb these processes and lead to both immediate effects as well as latent or delayed effects, with sex differences, obviously, due to the differences in natural hormones and their disruption. And these developmental hormonal and EDC actions are manifested as differences in neural circuitry, the phenotype and numbers of neurons, and the behaviors and the processes that are controlled by the brain. In my lab, we have a rat model for developmental EDC exposure. What we've been using are 2 different classes of EDCs that act through different hormone pathways. One of those is vinclozolin, which is a fungicide and is known to act through perturbations of androgenic signaling. And the other is a group of polychlorinated biphenyls, PCBs. The mixture that we use is called Arachlor 1221, and this is an estrogenic mixture of PCBs. The way we do the studies is we start with a pregnant rat. We expose her to one or the other of these EDCs or to the vehicle during her pregnancy, during specific developmental windows that are important for gonadal development and brain development. And then we look at changes, direct changes to the F1 generation, the exposed individuals. And at the bottom, I've listed some of the endpoints that we've looked at in those exposed individuals. And we've shown many changes to hormones and genes and epigenetic marks in the brain. However, today's session is really about inter and transgenerational effects. And so what I'm going to be talking about during my brief time today is those effects that are manifested on subsequent generations. This models what we're trying to do in the rat, but it shows what's going on in the human. So if you have a pregnant woman who we call the F0 generation, her child, the exposed fetus, is the F1 generation. The germ cells developing within the gonads are the F2 generation, that would be the grandchild. And any effects of hormones or EDCs on these developing germ cells, if they are heritable, if they induce epigenetic molecular changes, they can be manifested as changes to the F3 generation, which is entirely unexposed. These are the great-grandchildren and are the focus of much of my work, as well as work of other people who are in the field. Okay, so let me orient you to the model that we're using in the lab. This is our 6th-generational model. We call it 2 hits of EDCs 3 generations apart. And I'm going to talk you through it, so don't get scared. I know it's complicated, but at the top would be the F0 rat being exposed to the EDC. At the same time, her developing fetuses, the F1s, and the germ cells, the F2s, are being exposed. And when we continue breeding across generations, we breed either through the female lineage, a maternal lineage, or we breed through the males, a paternal lineage. And this is very important because there are sex differences in hormone actions on the developing brain, but there are also sex differences in how both hormones and EDCs can change germ cells, induce epigenetic marks, such as methylation, demethylation, and remethylation. And those are quite different in males and females. And so by separating out the maternal and the paternal lineages, we can follow those lines and compare differences in how the EDC effects are manifested. Now, here's where the second hit of EDCs come in. And what we're trying to do is model the 2-hit situation, which is used in many fields, but usually within a lifetime. So 2 hits are often used in models of carcinogenesis, where there's a first exposure but to some sort of insult, but a second exposure is needed to really further result in some sort of a disease phenotype. And what we're doing here that's entirely unique is giving a second hit of the EDCs across the generation. So the first hit is happening in the F0 dam. The second hit happens to the F3 females, either maternal lineage or the females who are being bred with the paternal lineage to generate subsequent generations. And our hypothesis that we tested here was that groups that are given the same EDC, so as I said, we either give an estrogenic EDC, Ericler 1221, the PCB, or we give an androgenic, anti-androgenic PCB, vinclozolin, and we're testing whether the nature of the first hit affects the response to the second hit and how that is manifested across future generations. Just as a reminder, along with looking at 2 different EDCs and their combination, and either they're given the same hit or a different hit here at the F3 generation, we are also looking at sex differences in outcomes, as well as these lineage-specific effects. Now let me turn to some of the data, and because I only have 20 minutes, I'm only going to show you a very small subset of the data, but they illustrate the points that I want to make, I think, very clearly. So let me orient you to this slide. What this shows from left to right are changes in body weight at birth. Body weight, we found, is a really good biomarker of exposures to both hormones and EDCs. And so I picked the day of birth as just an example of what body weight looks like in animals exposed to EDCs. Beginning with the F1 generation here on the left, these were directly exposed. And going from left to right, the groups here are the vehicle, DMSO, the Ericler 1221 group, and the vinclozolin group in females on the left and males on the right. And I think you can see that the PCB animals are slightly smaller, but significantly smaller in both males and females in this F1 generation. I should mention what the Ns are in this experiment. The Ns, the number of individuals per group, is between about 20 and 50 for all of the groups that I'm about to show you. So the Ns are really large, enabling us to really feel confident about these differences, which are highly significant. Now, as we start to move across the generations, I'm going to jump to the F3, the F4, and the F6 generations, because those are our focal generations. What I'd like to point out, without belaboring the details, because there are a lot of details, is that across the maternal lineage, there are very few effects of EDCs that are maintained in the F3 generation, and we also see very little going on in the F4 and the F6 generation, where the F4 had that second hit. However, the paternal lineage was quite profoundly affected across the generation, both in the females and the males, and you can see that in the F3 generation. And I'd like to point out in the F4 generation and the F6 generation, something that's both very important and very interesting. So just to, again, orient you to this complicated slide here, in the F4 generation, the groups shown here are animals that received 2 hits. Going from left to right, 2 hits of vehicle across generations. Purple is 2 hits of the PCBs. Lavender is the first hit being Arichlor, the PCB, and the second hit being switched to vinclozolin. That's a switch group. And that lavender group is the group that we're really interested in, because that switch group is different from groups that didn't get switches, as shown here in purple. And it also differs from groups that got 2 hits of vinclozolin here. And you can see that both in females and in males. And that is something that carries across to the F6 generation, where you can see that lavender group, the switch group that got first a PCB and then vinclozolin across the generations, was different from other groups. That same lavender switch group also is affected for behavior. So let me, oops, my slide, it does not want to go forward. Okay, so this shows age at puberty. And so these are the same animals. The slide is organized the same way. And you can see that there are some maternal effects, but I'm going to focus here on the F6 paternal generation, where you can see in the females and in the males that this switch group is also the one that is significantly different from all of the other groups in timing of puberty. So puberty is a little bit later in that switch group in the females as well as in the males. And the last piece of data I want to show you, because we're a behavior lab, and so I want to show you an example of one behavior that also illustrates how that switch group changes across generations. So here, what we did was allow the animals in all the generations to grow up, and we ran them through a battery of behavioral tests that included tests of social behavior, anxiety-like behaviors, and reproductive behaviors. And I'm going to focus on the anxiety-like behaviors, because we consistently see effects of prenatal EDCs on anxiety-like behaviors in the F1 generation. And this is something that we also see carry across to the F3 generation, and something that responds to a second hit in the F4 generation that carries across to the F6 generation. And again, it happens predominantly across the paternal generation. I should say there are absolutely effects in the maternal generations. We just see more effects in the paternal generations. And so that allows me to illustrate my point more clearly. But when we look at aspects of behavior on an open field test, where a rat runs around an open chamber, rats like to spend more time in the corners and along the walls, and they're more averse to spending time in the middle, where there's a little bit more risk potentially in their minds of things like predation. They're more exposed. And what we see is that when we look at something here, let me illustrate in the F4 paternal lineage for males and females, when we look at the males here for the time they spend in the corners and the time they spend freezing, which is a real index of anxiety-like behavior, we can see that there are differences between the groups. And again, that switch group that got the first hit of PCB and a second hit of vinclozolin is most different in those behaviors from the other groups. We also see changes in the females, and we also, if you look at the F6 generation, see some differences here a little bit more so in the females than the males. There's more variability, so, and the group sizes are between 18 and 29, so they're a little bit smaller. Okay, so to summarize this first part of my talk, the 2-hit 3 generations apart model. And I neglected to acknowledge David Cruz. David and I did all of this work together. He was my equal collaborator on this project, and I need to give him credit. So to summarize the results, the second EDC hit, given 3 generations later, elicited a novel phenotype. And the nature of that hit, whether it was same or different from the original hit, is a key determinant in the outcome. Now, let me point out that this is ecologically relevant because as new EDCs are coming onto the market, potentially with the same or different mechanisms, this is overlaid upon these historical heritable processes that our ancestors have encountered. Now, we also found the paternal lineage was disproportionately affected in the majority of the endpoints. Virtually every phenotypic outcome we looked at in all the generations were sexually dimorphic. Now, that's not surprising because we intentionally selected behaviors and traits that we predicted were sexually dimorphic because that's part of our initial hypotheses about EDCs having different effects in the sexes. Based on what we know about development, we also believe that EDCs affect regulatory genes or groups of functionally related genes that may result in significant effects on the adult phenotype. Finally, there must be some mechanism, presumably an epigenetic molecular mechanism, responsible for the heritability of these EDC effects in order for them to be assimilated into the germline. So let me turn to the next set of experiments that I'll go through very quickly that address this mechanistic question. So the important questions here are, how is information passed to future generations? Furthermore, how can a germline change be manifested in a somatic tissue like the brain and result in some sort of behavioral effect? To do this, the work done, again, in collaboration with David Cruz and with Ross Gillette's leadership, we did a 3-generational study. We just did this in, it's a little easier, we just did this in males. We just bred through the paternal lineage, and the focal generations in this study were the F1 generation and the F3 generation. Here, the animals, it was the same exposure model, and in the F1 and the F3 generation males, we collected sperm, and we also took 2 brain regions, CA3 of hippocampus and the central amygdala, which are involved in some of the behaviors that we find affected by the EDCs. And Ross looked at promoter methylation in the sperm in these 2 brain regions using RRBS, which is reduced representation by sulfide sequencing, which focuses on the CPG-rich DNA regions, the CPG islands. So our approach is a little different from some of the other labs doing work on DNA methylation. And I think our results are very complementary, or give complementary information to work focusing on other parts of the genome. This work was done on adult male rats. And I mentioned tissues came from sperm, CA3 of hippocampus, and central amygdala, which are important for the behaviors we study. And the amygdala in particular is highly involved in these anxiety-like behaviors. So let me quickly step you through the results. And this study is published. The previous study was not published, but these are published data. And what Ross did was make heat maps of the CpG sites that were either hypermethylated, shown in orange, or hypomethylated, shown in blue. And if you look at the numbers of genes that were affected, or number of sites that were affected, I should say, they differ between the different exposures, and these are all compared to their same generation vehicle groups. So for PCBs and vinclozolin in the F1 generation, there were about 200 to 250 sites that were differentially methylated, most hypermethylated. And we saw a similar pattern in the F3 generation caused by the two EDCs. And Ross did hierarchical clustering to look at the promoter methylation patterns amongst the different groups. And so this sorts the data into groups that are more highly similar in their methylation patterns by hyper and hypomethylation. And the way these data end up sorted in sperm is that they sort by generation. So the two F1 treatment groups, even though the treatments are different, the methylation patterns are more similar than they are to the F3 generation. And the same thing happens within the F3 generation in sperm. We get a very similar result in CA3 of hippocampus. There are fewer CpG sites that are identified. It's between about 75 and 150. Again, more of them are hypermethylated, and the groups sort by generation. The amygdala, which is important for behavior that we study, is different in its sorting pattern. So again, more genes are hypermethylated between about 100 and 150, and they sort by an interaction of generation and treatment, where the F3 group is different, and that's shown here with the bracket on the left, from the other three groups that sort together. So this tells us something about differential effects of PCBs across the generations. Ross went on to look at which genes were differentially methylated, and he made these cool gene clouds looking at unique, or which genes were differentially methylated, in this case on the left in the PCB treated this is in the F3 generation, in sperm, excuse me, in CA3 of hippocampus, and in the central amygdala. And then he looked at which genes were in common between the different tissues, and finally, which genes were in common in the middle amongst all three of the tissues. And he did this for both of the EDCs. And I think what you can see is this letter pRNA that is big in the middle of all of these gene clouds. These are promoter-associated RNAs. These play a role in silencing ribosomal RNA, so they play roles in translation, and they are all consistently hypermethylated in brain, both brain regions, and in sperm by both of the EDCs in the F3 generation. So to summarize, the general observations we made in this part of the study are that epigenetic modifications such as CpG methylation induced by EDCs are transmitted to the F3 generation, consistent with other studies that have been done in this domain. Each EDC has a unique DNA methylation pattern. In both sperm and hippocampus, groupings of CpG methylation are driven primarily by generation, whereas in the amygdala, the groupings are driven by EDC generation interactions. A little bit more specifically with respect to the promoter-associated RNAs, many of the CpG islands, I didn't show you this, but many of them were in intergenic regions associated with non-coding regions, of which 6 were in association with these promoter-associated RNAs, all of which were hypermethylated. And I should note that this hypermethylation would decrease gene expression of the promoter-associated RNAs. So this is my last slide, my conclusions and implications. Individuals of each generation experience perturbations throughout their life history. And these experiences, together with the ancestral epigenetic processes, shape their responses to current and future environmental challenges, something that we modeled in our 2-hit 3 generations apart study. The nature of the exposure, whether the chemicals act through similar or dissimilar mechanisms, determine the outcomes, again reflected in our model of exposure, where we have both an estrogenic and anti-androgenic EDC. Now, for this exposure to be transmitted across generations, it must induce some sort of programming of the germline, but it also must be incorporated into the target tissues responsible for the phenotypic outcome on brain and behavior. We also showed in sperm and brain that Erichlor 1221 and vinclozolin have unique signatures, and thus epigenetic modifications and the information that they encode lead to changes that require the transmission of, or the transfer of information to germline to somatic changes in the brain. So I'll end there and stop sharing my screen.
Great. Thank you very much, Dr. Gore. I think Jill has a question for you.
Yeah, thank you so much for that presentation. I just have a very quick question. I think that a lot of people are always asking about dosages, right, for these types of experiments and whether or not these things are physiologically relevant in humans. Mm-hmm. Of course, my major interest is in pharmaceuticals and hormone disrupting pharmaceuticals, which are administered in very, very high doses. But these environmental chemicals, at least in the general population, are only seen in low doses. What do you think that says about the implications for public health, owing to, generational exposures to environmental chemicals?
Yeah. So, well, I don't know if I mentioned my doses. I think they were on the slides. In our study, we give 1 milligram per kilogram over the course of 10 days during gestation, which is so we haven't measured how much actually gets to the brain or how much gets to the germ cells. But we're probably within exposure ranges of humans, probably on the high exposure side, but not outside the realm of the kinds of exposures people would have who eat fish out of the Great Lakes or the Hudson River, or are agricultural workers or live near agriculture where the fungicide is being used.
Got it. Thank you.
Great. Thank you very much, Dr. Gore. And I know we have some questions specifically asking about whether these treatments really affect the composition of different cells that make up these brain regions. And I think for the sake of time, we can just let that sort of play out by typing your answers and your responses in the Q&A session over Zoom.
Okay, excellent. I'll do that. Yeah.
Thank you. Excellent.